So first of all, thank you for the organizer that invited me for this conference, and I'm delighted to be here. Today, my topic is tick-borne diseases of cattle, understand pathogen transmission for the development of control strategy. I work for United States Department of Agriculture, Animal Disease Research Unit in Pullman in Washington. Just to make a location here, we are not in Washington, D.C., we are in Washington State. And the mission of our unit is to solve basic and applied problems concerning persistent infectious diseases of livestock, including horse, cattle, goats, and sheep. And we have 5 projects in our unit. Thank you. 5 projects: Scrapie project, respiratory disease for small ruminant. We have MCF, malignant catarrhal fever, Anaplasma marginale, anaplasmosis, and bovine babesiosis. My laboratory focuses on tick-borne pathogen of livestock, and we try to develop strategies to control tick-borne pathogen of cattle in the United States. And why is the United States? Because these parasites, especially Babesia bovis and Babesia bigemina, this disease is not present in the United States. However, the disease is very common in our neighbor country, that is Mexico. So pathogen transmitted by ticks vector remain the major challenge and a challenge concern to improvement of animal health, and especially for Babesia bovis, Babesia bigemina, and Anaplasma. And why it's so difficult to control these diseases is because when the parasite infected the mammalia host, they have a 2 infection phase. One is the acute infection that have a very high parasitemia, indicate here. And then if the immune system of the animal control the disease, they become persistent infected for life. And both phase, the acute infection and the chronic infection, is important for the ticks to acquire and transmit the disease to mammalia host. Today, I'm going to focus on bovine babesiosis. Bovine babesiosis is one of the most prevalent tick-borne diseases of livestock. It's distributed worldwide. For Babesia bovis and Babesia bigemina, they are distributed in the subtropical and tropical region, include Asia, Africa, Central and South America, and North America, in Mexico, Europe, and Australia. These parasites are associated with the distribution of the ticks vector, Rhipicephalus microplus. In contrast, a third parasite that causes bovine babesiosis is Babesia diversion. It's only distributed to Europe, and is also associated with the presence of the vector in European countries. Bovine babesiosis causes a significant economic loss for the livestock. For instance, last year in Latin America, more than $800 million have been lost caused by bovine babesiosis. And these losses are associated with a high morbidity, and during the acute infection, especially during the acute infection, with a high fever, ataxia, loss in weight and milk production, and also in some cases, they are high mortality. And these high mortality are associated with cerebral babesiosis caused by Babesia bovis. Abortion is common, and also this can, consequence of these diseases for the animals is the reduction in meat production. Babesia have a very complex life cycle. They need an intermediate and definitive host to replicate and transmit to naive animal. When the parasite infected the red blood cells, they replicate inside of the red blood cell by binary division. They cause acute disease in these animals, and the immune response control the parasitemia, and these animals become persistent infected for life. The ticks come and feed on these animals, and they ingest the parasites, and these parasites transform in sexual state in the lumen of the ticks. This tick here, female ticks, is able to ingest about 2.5 ml of blood from the animals. And then this parasite transform in sexual state, invade the middle gut epithelial cells, and transform in kinites. Kinites are released into the hemolymph of the ticks and invade the ovarian epithelial cells, and vertically transmit to the eggs. The next generation of the ticks are able to transmit the babesia to the naive animals, and these remain cycling for many, many years. So there are several control strategies to prevent the transmission of bovine babesiosis. Today I'm going to discuss with you vector control, development of vaccine to prevent disease and infection. I'm going to touch a little bit, maybe not drug therapy, because there is a presentation afternoon that he will focus on the drug treatment. I will focus also a little bit in the vaccine development to prevent transmission. Important also is the sanitation, hygiene, and education of the owners. So for the vector control, there are three important methods to control bovine babesiosis by control the ticks. One is the chemical acaricide, the treatment of the animals infested with the ticks, with organophosphorates, syntactic pyrethroids, and amitrax. However, in the past 5 or 10 years, Mexico have been identified population of the ticks that are resistant to single or multiple acaricides, and make so difficult to control the transmission of Babesia bovis to naive animals. There are vaccines that was available to control the infestation, the BM86 based vaccine. However, many countries have been used and have failed to control the infestation due to the variation of this BM86 protein in the gut. Our laboratory is focused on to identify molecules that can add to this vaccine to see if we can prevent the tick infestation. We found 2 molecules expressed by the salivary glands and the gut, aquaporin, that have been used in laboratory condition and show that there is a reduction of 25% of tick infestation. Also, we have a biological control by pasture rotation, and in this case, need to be a combination between the acaricide treatment and the vaccine for this biological control succeed. Without of this control together, the biological rotation or the pasture rotation will not work. Just to give an example that elimination of ticks also eliminate the bovine babesiosis. Here one very good example that have been done in the United States. We eliminate bovine babesiosis in the United States by control the ticks, Rhipicephalus microplus. And we eradicate these ticks in 1946. Now, the only place that we have these ticks in the U.S.-Mexico border. And all the animals that come from the Mexico need to be inspected and need to be acaricide treated. Here you can see that before they enter the United States, all the animals need to be treated with acaricide. If you find any ticks in these animals after the treatment, they will ship back, we ship back this cattle population to Mexico. We will not accept any animal that have ticks on the body of these animals. So to show how long it took for the United States to control bovine babesiosis, here is when we start the control strategy, the tick eradication program in 1906, and U.S. declared free of Rhipicephalus micropus in 1946. Took about 40 years, a heavily acaricide treatment with involvement with state veterinary, federal veterinarians, and the owners to eliminate, eradicate Rhipicephalus micropus from the United States. Today, we have only this area here that may have a little ticks, Rhipicephalus micropus. USDA established a quarantine zone that none of the animals in this area here can have the ticks on feed on the animals. All the animals that come from Mexico are treated, inspected, and treated before entering the United States. And this is the way that we try to keep the U.S. free of bovine babesiosis. However, there are alternate hosts to bring ticks from Mexico to the United States. USDA doesn't inspect deer or new guys to come from Mexico to the United States. Most of the ticks are freely to go to Mexico, and ticks can feed on these animals, attach to these animals, and these animals can come to the United States. However, they are poor hosts. However, there are a lot of the population have been increased in the last 5 years. One is because of the hunting of these animals in Texas, and the other is because of the preservation of the white-tailed deer by the wildlife. There is a new guy, another animal here that is very good to feed ticks on these animals as close as the cattle. And this animal also can go freely to Mexico and come back to the United States and can bring the ticks to the United States. And they can bring both susceptible and resistant population of the ticks. There are other exotic antelopes that also are responsible to bring some ticks to the United States. So as you can see here, there are several threats to the United States cattle industry. One, as I mentioned, there are now a lot of tick population resistant to acaricides. With the global warming, climate and land change use favor the reestablishment of the ticks in the United States. The alternate host that can bring the ticks to the United States, such as white-tailed deer and new guy. In addition of all these threats, United States import more than 1 million head of cattle per year from Mexico. Most of these animals are infected with Babesia bovis or Babesia bigemina. As you can see here, combination with the ticks in the United States and the introduction of infected cattle, we can have a catastrophic problem in the United States. And this can cause a shortage in the meat and milk production in the cattle industry. So our laboratory also focus on the development of vaccine to prevent disease or infection. And what we try to do first is to sequence the genomic of Babesia bovis. And when we sequenced here in 2007, we were able to compare Babesia bovis genomic with Thalelia parva and with Plasmodium falciparum. And these two have complete different life cycle within the vector. This is a mosquito, this is a intra-stadial transmitted parasite, and this is a transovarial transmitted parasite. It's very difficult to compare transmission of this parasite when they are different model of transmission. However, the genomic sequence help us to identify some genes that can be important for the vaccine development in the mammalian host. So what we have been done in the past many years, we try to identify genes that potentially can be used for vaccine development to prevent the infection of the red blood cells. We work with RAP1 proteins and BBOV RAP protein 68. And these two proteins are expressed in this complex, AP complex of the merozoites. You have the small open reed frames that is expressed by the merozoites of Babesia bovis. You have this BBOV57. This is an interesting molecule because in Theileria parva have been shown that this the syntonic genes is associated with the infection of a sporozoite to the lymphocyte of the mammalia host. We have this MSA1, major surface antigen 1, and major surface antigen 2. However, these two molecules are very variable between the strains of Babesia bovis worldwide. We have this VISA, this variant erythrocyte surface antigens, and these have more than 150 genes in the genome of Babesia bovis. These genes are associated with two things. One is to cause site-wide adhesion on the brain, the capillary, and second is because of a lot of genes variation in the family, they are able to invade the immune system and cause the persistent infection of the parasite within the mammalia host for life. We try to develop and test individual protein to see if they can control the infection or the diseases. We use individual RAP1, B. bovis RAP68. We immunize cattle with B. bovis, BOV57, and the others antigens. And then we test if they are able to control the infection or prevent the infection or the disease. Unfortunately, none of this molecule by itself control the infection or the disease. All the animals die in about 9 days post-challenge. The only vaccine that is very efficient is the live attenuate vaccine. However, there are several concerns about this live attenuate vaccine. First of all, the way that they make this vaccine is through a multiple inoculation in splenectomized animal. And during this process of making this vaccine, we may have some contamination with virus, with protozoa, other protozoa than Babesia bovis and Babesia bigemina, and also some Anaplasma marginale. Instead, we are controlled for Babesia bovis. We are introduced different pathogens in the herd. That is the biggest concern for the United States, and the United States do not allow the use of live attenuate vaccine in our animals. So all the animals that are born and raised in the United States, they are immunologically naive animals. So in addition, the live attenuate vaccine prevent the disease manifestation. However, they don't prevent the superinfection, means that the wild type parasite can infect the vaccinated animals. When the ticks feed on these vaccinated animals, they are able to acquire this wild type and they can transmit to naive animals. That is a big concern that the United States have to maintain this vaccine in the United States and cannot prevent this infection of the parasite through the ticks transmission. So again, what's the consequence of the introduction of bovine babesiosis in the United States? So again, there is no vaccine licensed in the United States, and all the vaccine that we test individually as a recombinant protein fail to protect the animals against the challenge. All the animals in the United States, they are immunologically naive because you don't have the disease circulate in our country. And then if there is an introduction of this disease in the United States, we will have a huge economic loss for the cattle industry. Because they are immune naive, up to 90% of adults infected animals will die in consequence of bovine babesiosis. So now I want to shift a little bit my talk and start to look at if you can develop a new strategy to control tick infection. And in this innovative approach, we try to develop or find molecules that is expressed by the parasite during the development in the middle gut or in the kinids. Here is the way that we are predicting that will work. We have the blood state, and then they form this piriform shape here. When the ticks ingest the parasite, they induce the transformation of gametes, male and female, and they fuse to form the zygote. And this induction is associated with the presence of enzyme, xanthurenic acid, and also when the ticks drop off, the difference of the temperature from the blood, that is about 37, 38 degrees Celsius, to about 26 degrees Celsius, induce the formation of this sexual state in the gut of the ticks. They fuse to form the zygote. Zygote invade the middle gut epithelial cells and transform kinids. Kinids then release to the hemolymph and then invade the ovary. They vertical transmit to the eggs, and the next generation of the ticks, the larvae, is infected with Babesia bovis, and when they feed on the animal, they will transmit Babesia bovis to naive animal. So, just to remind you, we are looking for genes that are expressed in these stages of Babesia bovis. What is the gap knowledge, or the knowledge gap in this research? First, we don't have a lot of proteins that is Babesia tick state-specific proteins. The proteins that is expressed exclusively during the development of Babesia bovis within the ticks. We don't know anything about the function of the ticks state-specific proteins, and there is no strategy to prevent tick infection with Babesia bovis. So what we did to identify some molecules specific to Babesia bovis kinids, we did some experiment to compare as a comparative proteomic, transcriptomics, and also with protein expression. So the design that you did here was inoculate the mammalia host with Babesia bovis stabulates. When the parasitemia start to get high, about 5%, we apply the ticks to feed on these animals and let the ticks to feed to engorgement. Then the ticks drop off. We collected the ticks, select the ticks that have a high level of kinids. This is the kinids, Babesia bovis kinids in the hemolymph of the ticks. And then we compare the expression of the protein between the blood stage and the tick state. So the first part of our experiment, we compare proteomic analysis. We use the side-eye to label the tick hemolymph, in this case side-eye 2. The side-eye 3 label the Babesia bovis kinids plus hemolymph, and side-eye 5 label only Babesia bovis blood stage. And each of these side-eyes have a different wavelength that we can separate during the process of 2D gels. And then we can identify genes that are important for expressed only by the kinids stage. And then we mix all these proteins labeled with these side-eyes, do the 2-gel electrophoresis, take the pictures, imagine here, with the analysis, select the spot, do the mass spec, mascot, and identify the protein. In this example here, we have in green, in tick hemocytes, and in red, ticks, Babesia bovis kinites. And as you can see here, you can well see that the green are specific for the ticks and the red is specific for the kinites. To give a better example for how we did this experiment, we compare the first one, kinites, hemocytes, with blood state. We want to determine which protein is different expressed in the kinites versus the blood. Here, for instance, you see in red, indicate that probably is the kinites, but there is a possibility here that this protein also can come from the hemocytes. So the green is blood state, the red is the kinites hemocytes. And then we compare kinites hemocytes to only hemocytes. Hemocytes in green and kinites hemocytes in red. If they are green, they should show on the areas that are only hemocytes, and the one that is kinites is in red. Here again, show that they are only expressed by the kinites hemocytes. We are very confident that this protein here are kinite-specific proteins. And then we compare the blood state with the hemocytes, and there is no signal either for blood state or for hemocytes that confirm that these two proteins are kinite state-specific proteins. Then we look at the gel to see which spot have this protein. Here, again, the spot for Babesia bovis kinite hemocytes. And when you look at the tick hemocytes, you don't see any protein here, and neither in the blood state. And then we look at the histogram showing the peak volume of protein. As you can see here, you see the peak, nice peak of Babesia bovis kinite hemocytes. There is no peak in the histogram here shown in Babesia tick hemocytes, and neither in Babesia blood state. As you can see here, these are proteins from the ticks. Very clear that both preps, the Babesia bovis kinite hemocytes have, and only the ticks hemocytes have. We are very confident that this protein here is indeed a state-specific protein for the kinites, Babesia bovis kinites. We select about 50 protein, but I'm gonna just show a few proteins here for you. And one of the protein very interesting is this 2220 with total iron score, confidence interval over 95%, and is hypothetical protein. A second protein that we found in this approach is 6250, is a GCC2, GCC3 domain contained protein. Is very close related with malaria parasites. And here are the rest of the list that show that they are conserved hypothetical proteins. We also do some in silico analysis to determine if these have a signal peptides, transmembrane protein, transmembrane domain, if they are ortholog to the other parasite, tick-borne, and in case here, our outlier is Plasmodium. As you can see here, the 2220 have signal peptide and transmembrane, and the predicted protein is about 70 kilodalton protein. Interesting that we find here is that this protein is ortholog only in Babesia bigemina, and the rest, they are not expressed or have these genes in Babesia diversion, Microti, Teleria annulata, Teleria equi, Teleria parva, and Plasmodium. And these two parasites, Babesia bigemina and Babesia bovis, they are transovarial transmitted. The others are not transovarial transmitted. And that we are thinking that these proteins perhaps are associated with the attachment or invasion of the ovary epithelial cells. You have this 6250, That is, there is no signal peptide, there is transmembrane, and is expressed in all, especially in this plasmodium that have the DCC domain. So we want to confirm our find by determining the differential transcription expression between the blood stage and the kinase states. We have here blood states that we isolated, the RNA and also the kinase. We extract the total RNA, synthesize cDNA with 109 nanograms of the total RNA. We did the serial dilution of the cDNA to determine how the concentrates, the different expression, transcription level between the blood state and the kinase. We did the PCR. We detected the amplicon by 2% agar-delta, and we sequenced the amplicon to verify that the genes that we are looking are indeed the specific genes for Babesia bovis and Babesia bigemina. So let me walk with you the result here. You have the Babesia bovis here, you have the kinase cDNA and the blood cDNA. We have the genes here, and we want to determine the difference expression between the two states. We have the dilution 1 to 10, 1 to 100, and go to 1 to a million. And here's the same. So as you can see here, we see that 20 to 20 have a high transcription level as compared to the blood state. In 60 to 50 is also expressed in the blood, in the kinase, higher than in the blood. Interesting for us is that the rest of the list here, they are more expressed in the Babesia blood state and less in the kinase. We also compare these finds with the orthologs of the proteins with Babesia bigemina. We repeat with this the same setup here in Babesia bovis we did with Babesia bigemina. With kinase, with blood state, and we use the ortholog protein primers to the ortholog genes to 20 to 20, 60 to 50, and as you can see here, the level of the transcription for this ortholog is higher in the kinase and low in the blood, as well as 60 to 50 high in the blood and low in the high in kinase and low in the blood. And here mirror exactly the same what we found in the Babesia bovis blood state, high in blood state and low in kinase. And these GCC2 and 3 are domain-contained protein. They are orthologs to malaria. And you will see that this protein have a very interesting function. And there are 4 proteins for GCC2 in malaria, PCRMP1 and 2, that invade the erythrocytes, have a PCRMP3 and 4, invade multinucleate cells, salivary glands of the mosquito, and the hepatocytes of the human. Here, we look at this ortholog for this protein to Babesia bovis, and we find 4 GCC2, GCC3 protein domain in the genome of Babesia bovis. Then, we compare the structure of this protein, the GCC domains, to between Babesia and Plasmodium. And as you can see here, they have this domain here, the GCC2 and 3, and also they have this EGF-like domain. Have a large protein, both in Plasmodium and Babesia, and also this transmembrane protein. Now we want to determine if there is some relationship between the expression of the transcript between the blood state and the kinase state of Babesia bovis. So what we did here, we again did the cDNA for the kinates and the blood state, and used all these primers to amplify specifically 6250, 11730, 11740, and 6260. As you can see here, these two genes here is highly expressed in the kinates and low expressed in the blood. In contrast, these two genes here, they are highly expressed in the blood state and low expressed in the kinate states. We are very confident that these genes perhaps are associated with the invasion of the ticks, and the others are associated with the invasion of the red blood cells. So then we want to determine the protein expression of this protein on the babesia bovine blood state and kinate state. We need to generate a polyclonal antibodies in rabbit, and the criteria that you use to select the protein is the protein needs to be detected in the 2D gels, and the detection need to be blood state, babesia bovis state-specific transcript, and they need to be conserved among the arthropod-borne pathogens. Then we design these peptides, 3 peptides based on in the B cells epitope. If these peptides are surface exposed protein, we conjugate with KL8, and then we immunize the rabbits. So to determine if this protein is expressed on exclusively in the kinate, we prepare a smear for babesia bovis blood states, kinate states, and we did reactivity with rabbit anti-kinase antibodies. Use a secondary antibody, anti-rabbit IgG, FITC, and you analyze the reactivity by confocal microscopy. We analyze all the proteins that we are interested, but today I'm going to just show what's happened with this 2220 that have ortholog function with the babesia bovis, babesia bigemina protein. All the pre-immunosure from all the immunized rabbit have no reactivity with blood state or kinates. And when you look at the reactivity of the antibodies, rabbit antibodies against the blood state, we have no reactivity. In contrast, the kinates have a huge reactivity with kinate states. Here show the babesia bovine nucleus labeled with DAPI in blue. And here show the reactivity of anti-peptides antibodies. Very clearly that these proteins is expressed only in babesia bovine blood states, kinate state, not in the blood state. Here's the merge image. You can see the blood and the nucleus, and you can see the reactivity of the antibody. Here you just see the nucleus. So the conclusion for our finding in this part is we identify babesia bovis state-specific protein. And this state-specific protein can be a potential target for the development of transmission block strategy. We found that 2220 and the homologs, orthologs protein may be responsible for kinates attachment or invasion of the ovary epithelial cell prior to do the transovarial transmission. We also found that 6250, 11730, and 5840 are expressed by the kinates perhaps for the parasite adaptation with the ticks microenvironment. With that, I would like to acknowledge several people that make this research possible. Dr. Don Knowles, Guy Palmer, Carlos Soares, Glenn Scholes, Naomi Taus, and Hala Elziar, and the support of our researcher, support scientists, Carol Johnson and David Hernan, and the people that take care of our animals. Ralph Horn, James Allison, and Kat Mason. And this project has been supported by USDA ARS, that is the internal grant, and our USDA NIFA grant. Thank you for your attention, and I'm glad to take some questions.
Okay, thank you very much for Dr. Ueti. ありがとう、Ueti 先生。